X-ray diffraction method produced data by three major scientists like Maris Wilkins and Rosalind Franklin proposed a very simple, famous double helical structure for the DNA. One of the hallmark of their propositions was base pairing in between the two strands of polynucleotide chains. However, this phenomena was proposed based on the observations of a scientist called Irwin Chargaff. That for a double-stranded DNA, the ratio between adenine and thymine, guanine and cytosine are constant, are equal ones. Okay, very important point. Ratios, as per the Chargaff rule, ratios between. So I have posted some of the questions related to DNA. Please look at them. So you will understand the ratios in between the various sugars. Adenine, ratio in between the adenine and thymine, guanine and cytosine. Okay, are constant in equal ratios. The base pairing confers a very unique property of the DNA or genetic material to the polynucleotide chains. They are said to be complementary to each other. Okay, and let us take some predictions here. If each strand from a DNA act as a template for synthesis, if we take the DNA as a double helical structure, let us take one strand of the DNA, it act as a template. Okay, na? What is that? Template. What is the use of template? In the coming videos I will tell you. But it act as a basic structure for synthesis of new strand of DNA. It act as a Xerox machine. Template means Xerox machine. Simply we can say that. The two or double stranded DNA, if you carefully see, okay, thus produced would be identical to parental DNA or parentical DNA molecules. Parent DNA molecules. Okay. Apart from this, because of this, genetic implication of the structure of DNA become very clear. The silent features of double helical structure of DNA, what are they? First one, it is made up of two polynucleotide chains. Second point, they act as a backbone of the DNA. Two main chains have anti-parallel condition. Okay, their polarity, especially their arrangement, their polarity is anti-parallel. It means one chain has, if you take the one chain, it has polarity of 5 to 3 end, okay, 5 to 3 end, whereas another chain will have 3 to 5 end condition. In this case, in the diagram also I will show you. And the third character is the bases in two strands. If you take two strands have bases paired through hydrogen bonds forming base pairs. Okay, adenine forms two hydrogen bonds with thymine from the opposite strand. Only adenine forms two hydrogen bonds. Similarly, if you take the guanine, another interesting character is guanine, if you see, is bonded with the cytosine, with 3 hydrogen bonds. Very important point. And as a result, always purine comes opposite to pyrimidine because of this condition. This generation or this generates approximately the uniform distance between the two strands of helix. Now, the two chains are coiled right-handed fashion. So the two strands are coiled together in a right-handed fashion. In case, if you take the pitch, what do you mean by pitch? You know very well about the pitch of the helix. Okay, let us take this is a helix. The pitch of the helix, if you carefully see, pitch of the helix. The pitch of the helix is 3.4 how much it is? 3.4 nanometers. A nanometer is 1 billionth of a meter. And there are roughly 10 base pairs. How many? 10 base pairs are there in each ton. If you take in each ton, 10 base pairs are present in the DNA. You can see double stranded polynucleotide chain here. Adenine, thymine, guanine, cytosine. And adenine will always come opposite to thymine. Thymine will always come opposite to adenine. Guanine opposite to cytosine. Cytosine opposite to thymine. This causes these bases, if you take nitrogen bases, okay, they are present opposite to each other in the double stranded of DNA, and hydrogen bond is present in between these two bases. You can see in the diagram. Okay, cytosine forms 3 hydrogen bonds when it connects to the guanine. Understood, everyone? And apart from this, distance between the base pair in helix, it is approximately 0.34 nanometers. Okay, the plane of one base pair stands over other one. The plane of one base pair stands over another one. This addition to hydrogen bonds confers the stability of the helical structure. Okay, the stability of the double helical structure is depending upon the hydrogen bonds. Compare the structure of purines and pyrimidines. The proposition of, if you carefully see the double helical structure of DNA and its simplicity, explaining the genetic implications become revolutionary. Very soon, Francis Crick proposed central dogma for the molecular biology, which states that genetic information flows from DNA to RNA to protein, DNA to RNA to protein, which plays an important role in, you can see here in the double helical structure of DNA, they are present in the right-handed fashion. Each helix, you can see, arranged in the right-handed fashion. Okay, adenine and thymine, guanine and cytosine, you can clearly see here. And after this,